Hello everyone, a very warm welcome to the students of Dr. B.R. Ambedkar Open University, Department of Zoology, B.Sc first year, semester 2. Today's topic on this video session will be type study of Herdmania. Now, coming to the chordates, because after the invertebrates, we are stepping into the phylum Chordata. So, before going into the true characters of chordates, we have to talk about the three phyla that is present before the true chordates, that is phylum Chordata. We will be talking about three subphylums. Subphylum first one comes Hemichordata, then Urochordata and then Cephalochordata. The Hemichordata and the Cephalochordatas are the earlier primitives, and in between is the Urochordates. Though hemichordates we once thought that they were chordates, but recent study shows that they no more belong to the chordates. And then when we are talking about cephalochordates resembles the true chordata, and in between these two is the urochordates, or we also call them as tunicates. So, under subphylum they are called as tunicates. So, today's study we will be talking completely about urochordates, and the example under urochordates is type study of Herdmania. Now, before going into the type study of Herdmania, we will talk a little bit about what are these urochordatas. Urochordates are nothing but the tunicates, because earlier though it was called as urochordates, now it is known as tunicates. Why? Because the body is covered by a test called tunicata, the test or tunicates. Now, what are these tunicates? Tunicates are all invertebrates in adult form, though they come under phylum Chordata, but in the adult ones the chordate characters are disintegrated, whereas in the larval forms you find a proper chordate characters. Now, what is a proper chordate characters in the larval form? That is, it has a proper notochord and you have a dorsal neural cord, and then it also having the gill slits. So, all these properties show that the urochordates were once it also has affinity towards chordata, but as it moves into metamorphosize into the adult stage, it loses all its characters of the urochordates and they are under the invertebrates. So, this is the introduction of the tunicates or urochordates. So, and also what exactly they are, you always find them in the marine oceans, and these are usually called as sea squirts. Squirts because they squirt out the water from the siphons, that is it has two in-current siphons. One in-current siphon which takes the water inside, and there is another siphon that is a out current where the water is expelled out. So, because it expels the water in a squirt formation, it is called as sea squirts. Now, the body of the adult tunicates is quite simple, being essentially a sac or bag like structures filled with lots of water in them, because as I said it is having two siphons, one is a dorsal and one in the ventral side over there, the anterior and the dorsal side, and this both the siphons acts in to take in the water current and to throw out the water. Now, by doing so, these also are usually filter feeders. What do you mean by filter feeders? In these siphons, they have these tentacles which filters the water, thereby does not allow the heavy metals or heavier bodies to enter inside. So, these are usually, as I said, they are all microscopic feeders, that is they usually eat the microorganisms, the zooplanktons and algae and so on. And this urochordates, majority of them are sedentary because they do not move at all, and some are pelagic. They also have the budding also is found in this tunicates, and these are formed in colonies. The life history passes through a dramatic change, that is, as I told you all, the chordate character of the larval form completely changes into an invertebrate characters in the adult form, but this is not so in the cephalochordates. Now, coming to the classification of this tunicates, belongs to kingdom Animalia, phylum Chordata, subphylum tunicates. As I told you, this can also be called as urochordates. Class Acidaceae, because they come under Acidias. Order Pleurogona. Family Paiuridae. Genus Herdmania. So, So, this is the picture, if you can see in the picture. So, this is the structure of the Herdmania, and as I said, there are two apertures over there, the branchial aperture, the test of the tunic, and the atrial apertures. And the the body is completely sac-like structures which is filled with water, and then it has a narrow foot over there. So, this is the picture of how an Herdmania looks like. What is a habit and habitat? As I said, these are solitary and they are referred to as red-throated ascidians, because if you can see the picture, there is two siphons over here. Why we are calling them as red-throated ascidians is because they are red in color. Now, why they are red in color, I will be talking about that in the coming slides. So, it is exclusively marine, and the genus is recorded to be somewhere up till 9 meters to 21.6 meters inside the deep sea. So, usually they are found in the Indian Oceans and some a little bit at the Pacific Oceans too. They are sessile and always fixed to the base by the foot, and microscopic feeders. The tunic of Herdmania, the tunic, that is the covering of the Herdmania, that is called the tunic, provides shelter for many organisms. What do you mean by shelter for many organisms? If you can see in the second picture over there, the picture shows the Herdmania is covered by some algal growth, and there are some also microscopic organism which take shelter over this tunic over there. So, that is the reason why they provide shelter for many organisms, and the whole animal is completely covered. So, it hides within the algal of this growth over the tunics. Now, what is the external features? The body of Herdmania is roughly oblong in shape, completely oblong, narrower at the base because it is attached by a foot over there, and at the free end, as I said, it is provided by two external openings, or also called as the branchial and the atrial apertures. The average size can be somewhere around 9.5 centimeters height with 7 meters broad in length, and then 4 centimeters thickness. So, usually you find at 9.5 centimeters long, but in a completely grown adult you can also find them at around 12 to 13 centimeters long. So, the sizes can vary, but usually you find somewhere between 9.5 centimeters. The foot when present may attain a length of at least 3 to 4 centimeters when you can find the foot, that is in an area where there is sandy places. The foot can be seen as a narrow length, but when it is attached to a solid substratum, the foot is spread out. So, usually you don't see the foot of the Herdmania when it is completely attached. And also, as I said, why the Herdmania is red in color. Now, if you can see in this picture, when you can concentrate on these pictures, there is some red dots that you find on the siphons of the Herdmania over here. Now, what is this? This is nothing but the terminal knobs, or it is also called as ampullae in the blood vessels of the test, which is a characteristic feature of the Herdmania. So, the blood vessels they end in a form of terminal knobs, and this terminal knob is nothing but red in color. So, that is the color that is specific to this Herdmania. Now, if you can see the body division is what we have to concentrate over here in this picture. If you can see, as I said, there are two apertures over here. You have one branchial aperture and one atrial aperture over here. Now, branchial aperture is to the anterior side. So, wherever the branchial aperture is present, that side is called as an anterior end, and opposite to the anterior end is, you call them as posterior end. Now, towards the opposite of the branchial aperture, you have an atrial aperture. Now, this atrial aperture, the position of the atrial aperture is nothing but the dorsal side, and opposite to the dorsal aperture, you have the ventral side. So, the body is divided into 4 different parts, that is the anterior, posterior, dorsal, ventral. So, wherever the branchial aperture is present, that is called your anterior side, and opposite to that anterior side on the ventrally, it is the posterior side, and wherever you find the atrial aperture, that is called the dorsal side, and opposite to the dorsal is the ventral side of the Herdmania. And as I said, you can see the foot also, it is a little bit narrow, and during elongation it can go up to 3 to 4 centimeters. And also if you can see, the body is covered by this test like structure, the rough test like structures over here, and this is called the test or tunic. That is the reason why they are called as tunicates. And then inside you can see the body proper, the position of the body which is present inside. Now, the branchial and the atrial apertures are on the short protuberance of the body, and they are also called as branchial and the atrial siphons. Why siphons? Because it is taking in water and giving out the water. So, when fully extended, you can see that the atrial aperture is little bit longer than the branchial apertures, only in the length. But when they are open completely, when they open up, when the siphons are opening up, the branchial aperture is quite larger than the atrial aperture. So, only in the lengthwise you find the atrial aperture is longer, but when the siphons are opened up, when the mouth opens up, the branchial aperture mouth is quite larger than the atrial apertures. And also the atrial aperture is always erect or straight in structure, whereas the branchial aperture is a little bit bent to one side, and which is opening away from the atrial side. So, you always find the branchial aperture a little bit bent and away from the atrial apertures. And as I said, the centimeters, they are wide, 2 centimeters wide opening, whereas the atrial aperture is little bit lesser than the branchial apertures. Now, at the base of the branchial aperture, now we will go a little bit into what is happening inside and how the structure of this siphons are present within the animal. And before that, let me also tell you that the body, if you can see in this picture, the body is completely covered with the pink layer, is called the tunic or the test layer, what you call them as, and in the anterior side you can find the test is being completely covered within the siphons of both the atrial and the branchial siphons. And also the inside the tunic is the mantle, that is the Herdmania is covered outerwards as a test or tunic, and inside is called as a mantle covering, because it is covering the whole part of the body. Then if you can see the branchial aperture or the branchial aperture, that is what you can see over here. Now, this is entering inside, and this long elongated one is called a siphon, and within the siphon there are some tentacles that is present over here. Now, this tentacles are the ones which is going to filter the food that is coming from the water. So, as they are taking in the water, along with the water you can find some microorganisms also entering, and along with the microorganisms you can also expect some larger particles also trying to enter. It can be sand particles, it can be any large particles that can enter, but the filters, the tentacles that are present in the siphon area, that will not allow or captures the larger particles and throws them out, and it can allow only the microorganism to enter inside the Herdmanian body. So, you can see as they enter inside, there is a large pharynx that is present inside over here. So, this large pharynx is always completely innervated with stigmata, or it is also called as gill slits. So, this is where the taking in of the microorganisms or the absorption of the water and throwing of the water occurs in this stigmata. And then you can see the in the atrial siphon you do not find that much of tentacles what you find in the branchial aperture. So, because branchial aperture is nothing but the oral aperture or the mouth of the Herdmania. And what is atrial aperture? Atrial aperture is only used for removal of the undigested food, or the water that is taken in has to be thrown out. So, everything has to be done in the atrial aperture, atriopore. So, you do not find much of the tentacles, although the tentacles are present, it is not well developed, they are very smaller than when compared to the branchial aperture tentacles. So, the branchial aperture tentacles is very well developed and you have almost 62 branches of tentacles, because you have 8 tentacles, and within that 8 tentacles they are subdivided and subdivided into smaller ones. So, it acts somewhat like a sieving material, that is where you use the sieve to filter some juices or liquids. The same way the tentacles work for this Herdmania, wherein they completely filter the water that is coming inside, allowing only the microorganism and throwing out the larger particles. So, and then so that much of tentacle is not necessary at the atriopore. So, hence the tentacles are not well developed at the atriopore. And then as I said you, as the atriopore and the branchial aperture is also covered by this tunicates, because the tunicates, though it is covering only the outer part of the body, it is also covering a little bit inside the siphons also. Both the siphons is covered by this tunicates or the test cells. Now, why is the cell covering even the branchial and the atrial aperture? Is because if there is any slight disturbance in the water, if there is a heavy current of water coming, or if there is too much of dust occurring, and any slight disturbances, the test of the Herdmania tries to close the apertures immediately, thereby not allowing any particles to enter into this siphons. So, the test material acts also as a covering which does not allow the particles to enter inside, thereby closing tightly without allowing anything to enter inside. Now, coming to test. Now, we were talking a lot about the test. Now, what is this test and what it is made up of? It is nothing but it is an external covering of the Herdmania. So, that is called as test, or as I said in the beginning, it is also called as tunicate, because it is having the test or tunic. The other name of test is tunic. So, tunicates. This is usually leathery, translucent, and it is also composed of tunicin, that the protein name is tunicin. So, hence they are called as tunicates, and this is a substance which is almost resembling like that of the cellulose of the plant. So, imagine the cellulose of the plant, a little bit harder material that you find on the plants. The same way the test is also made up of the tunicin protein, which is leathery and it is also translucent. And the foot is entirely made of test, completely it is made up of test, and it acts as a receptor as well as respiratory organ. So, as I said, the respiration also has to occur, that is the taking in of the oxygen and releasing of the carbon dioxide has to occur. So, the foot also, because it is covered by a test, this test acts as a respiratory organ and also as a receptors for different sense organs. Now, what is this test composed of? The test is composed of a clear matrix, because it is made up of a matrix. It also has different various cells that are present, which is also various shapes. Then it has a interlacing fibrils, because it has to give some shape. So, it has interlacing fibrils, calcareous spicules to give the shape. It also has some calcareous spicules and also branching vascular vessels, because it has to have the circulation part also is necessary. So, it also has a branching vascular vessels. Now, what is this matrix made up of? Matrix is nothing but a gelatinous substance made up of polysaccharide called tunicates or tunicin. So, that is what it means, tunicins, because that is the part that is present over there. So, it is gelatinous, gel like substance, and it is made up of polysaccharide called tunicin. Cells, what are the different cells? So, it has different 6 to 7 different shapes and sizes of the cell. So, some are large eosinophils. If you can see in this picture, you can find there are some different cells over here. There are some large eosinophils, then some small eosinophils, then you have some amoeboid cells because it is not completely, it has a pseudopodium over there. So, it is also called amoeboid cells. Then you have some vacuolated cells over here, and also some nucleated cells which is having a large nucleus, and of course, some nerve cells with several processes that because the nerve cells can be bipolar or multipolar in Herdmania. So, you have some different cells over there, and it is also made up of squamous epithelial cells, the flat squamous epithelial cell which is made up which makes the tunic or the test material to have that type of particular shape. And then it is, as I said, it is having also interlacing fibrils. Now, this interlacing fibril is nothing but it is made up of some smooth muscle fibers, a fine network of smooth muscle fiber that you find on the test. Calcareous spicules. What is this calcareous spicules? There are two types of calcareous spicules that you find in Herdmania, and they are placed at equidistant inland, and also the spicules are having some rings. So, if you can see, as I said, you have two types of spicules. One is microsclers and the other one is megasclers. Now, if you can see this picture, the small one over here, the pin like structures over here is the microsclers which is found only in the test, and each consists of a round knob like head and an elongated tapering body. Now, this elongated tapering body has concentric rings over it, and you find some 5 to 20 rings that are present on this spicules, and the length can vary anywhere from 50 microns, and sometimes it can go up till 80 microns. So, this is the microsclers that is found only on the test. Now, the under the megasclers, you have two different types of megasclers over here. One is the spindle shaped megascler, and the other one is a this is a pipette shaped megascler, because it resembles like that of a pipette. It looks like a pipette. So, pipette shaped spicules, and this is a spindle shaped spicules. Now, the spindle shapes and the pipette shaped spicules also has some concentric rings. The rings can be more than that of the microsclers. It can go beyond 30 to 40 and it has it is always, the shape always resembles on the upside, the spicule resembles onto the top like that. So, the spicules are round on this the concentric rings are always facing upwards on the spicules. Now, where do you find this spindle shapes and the pipette shaped spicules? So, usually the spindle shaped spicules are found in the stomach and the gonads and also at the bases of the siphons. So, this usually you find in the region of the stomach, in the region of the gonads, and also at the bases of the siphons, and some are also present in the intestine, whereas the spindle shaped ones, the pipette shaped ones that you find over here, these are usually found abundantly in the mantle region, especially in the regions of gonads, and also in the lobes of the liver you find this pipette shaped spicules. Now, why the spicules? As I said, the spicules also give some steadiness to the test and the steadiness to the tissues or the organs. So, that is why these calcareous spicules are present on the body. Then, as I said, it is also having the blood vessels. Now, in the beginning we saw that the siphons has that red spots all around. This red spot is nothing but the blood vessels which terminate in a form of ampules, the terminal knob or ampules that you find. So, it is having extensive. Now, compared to other ascidians, Herdmania has a well developed blood vessels, and it has too much of vessels, blood vessels that is present inside when you compare with the other ascidians. And it, as I said, it is having a proper network in the test, and they end like the bulbs. So, if you can see in this picture, this is what you can see over here. So, these are the blood vessels, and this blood vessels terminate in the form of terminal knobs. These terminal knobs are also nothing but ampullae that you call, and these knobs are the ones which gives that specific red color to the test of the Herdmania. So, this is the particular meshwork of blood vessels that you find, and they also act as respiratory organs. So, these are the accessory respiratory organs, and the ampullae also acts as receptors of the organs. Now, morphology of Herdmania. So, we will try to see how they look like from the outside, though we have talked a lot about it, because a little bit of the morphology of the Herdmania coming to the body wall. The body wall of the Herdmania is called as mantle. Now, as I said, it is covered by a test, and within the test you have the mantle which covers the whole part of the body. The mantle is thick and muscular at the anterior dorsal region. So, if you can see in the picture, this is the anterior dorsal region, and this at this anterior dorsal region you have the mantle in a little bit thick and muscular, whereas at the posterior ventral region it is non-muscular and transparent. So, you can see some of the body parts also clearly visible from the ventral side of the body over there. So, at the anterior side it is thick and muscular, and at the posterior side it is a little bit transparent, and this transparent one is the tunic, and behind the tunic is a mantle. So, this mantle secretes the tunic or the test cells for the Herdmania. So, mantle is the one that secretes the test for the Herdmania, and the test is also attached to the branchial and the atrial siphons also. So, a little bit inside it is being attached. Why they are attached? It helps in the whenever there is any slight disturbance, as I said you, it aids in closure, immediate closure of the siphons. So, this tunic also helps in aiding the closure of the siphons immediately. So, thereby protecting the animal from heavy current of water or any heavy disturbances. Now, the general anatomy. Now, what inside, a little bit inside on the general anatomy, what we will be talking about, it is the body is roughly divided into two parts. If you can see in this picture, the body is roughly divided into two parts, that is the body proper. The first part is the body proper, which projects into the sea water, and below is the foot which lies embedded at the bottom of the sea. So, the whole thing is a body proper, and the foot is immersed inside the bottom of the sea. And if you can see the body, if you can clearly see, so this picture depicts the left side of the body, and if you can see there are two projections. One is the branchial aperture, one is the atrial aperture, and in these apertures you also find that they are surrounded by annular longitudinal muscles, and also annular muscles, and also the longitudinal muscles. So, this is the annular muscles you can find on either side of the apertures, the annular muscles, and there are some longitudinal muscles, and which are guarded completely by these muscles over there, and the siphons are also guarded by four lips you can see over here. So, this is the four lips where the siphons are being guarded. So, the projections which is having the annular and longitudinal muscles. So, this is the muscles, and also you can see you have the left gonad, the visceral side, you have the left gonad, then also you have the left liver, the simple liver over here is the left liver, then you have the stomach completely forming a U-shaped bulbous or large stomach that you find over here, and then terminating into intestine. Intestine forms a loop which resembles like which is placed parallelly around the gonads over there. So, this is the intestine, and the intestine opens into the cloaca like that. So, this is the left side of the Herdmania, and then we will talk about how the right side looks like. Now, in the right side, as I said, you can clearly see the intestine, the rectum, and anus part, and a part of the left lobe of the liver is also present. So, this is again the picture of how the left side is looking, that is again I am talking about the gonads and the stomach that you can see over here. So, you have the left side of the liver again, the stomach, the left gonad, and the intestine present over here, and of course, the annular and the longitudinal links.